so much for having me. I'm delighted to be presenting on the scientific end of the conversations we've been having about bioprinting and biofabrication. Today I'm presenting some research that I did as part of my exchange program. So I've got a background in physics, but I did the dual master's degree in biofabrication run between QUT here and also the University of Würzburg in Germany. So this bioprinting research was done while I was over in Germany last year. And it's work to do with the printability of shear thinning hydrogels, and I'll talk a little bit about what that means in a minute, for 3D bioprinting, and ultimately our application was for cartilage regeneration. But my research is in very general terms and looking at the development of all of these different hydrogels for a range of different bioprinting applications. And so we've just published these particular results in a paper in the Biofabrication journal, which is awesome, and we're hoping that this is going to be able to help labs all around the world who are developing bioinks for bioprinting to be able to speed up the process a little bit. So just a couple of definitions. Here we have some polymers, and when we put this into water, we call this an ink. And so this is a polymer solution. So these polymers can either be synthetic or natural polymers that we will use for our inks for bioprinting. And then when we add cells to these, this is when we call it a bioink. So a bioink is when our ink now contains cells. Just in case we're a little bit confused. And then we can cross-link this. And so this is a process that involves turning this polymer solution into a network where all of the polymer chains start holding hands, and it can improve the rigidity of the material. So this is when we're turning the liquid or gel-like bioink into something that's much more mechanically robust for our bioprinting applications. And so this is when we start to call it a hydrogel once it's being cross-linked. So we can do this with UV light or adding different chemicals as well. So hydrogels and bioinks are obviously very popular, not just in biofabrication, but they've also have been developed for cell culture and tissue engineering. I've also added these landmark review articles that talk about their applications in pharmaceuticals, and then of course more recently applying them to biofabrication and fabricating them using 3D printing that we're now calling 3D bioprinting specifically. So here's a couple of great examples from the literature where we've had a lot of conversations about 3D printing ears, and here's a good example of where they've created an ear cartilage structure with a particular hydrogel, or also looking at fabricating heart valves using a variety of different bioinks. So let's look at a bioink, and what do we want when we're developing a new bioink for a particular application, and there's a couple of really key criteria. The first one, it needs to be biocompatible, so this means it needs to be able to sustain cells and not kill them. Very simply, we want to make sure that we can put cells into that hydrogel and they stay alive and grow and proliferate into the tissue that we want. Second one is they need to be mechanically robust. There's absolutely no point 3D printing a bone made out of a soft, squishy hydrogel that's got the consistency of jelly, because we're never going to want to put that inside a patient. We're going to need something that's far more mechanically robust. So this is a consideration when developing hydrogels, that we are able to make something that's got the right mechanical properties for our final goal. And the last one is printable, and so this is particularly interesting once we start 3D printing our hydrogels. We want something that's able to be processed by a 3D printer and not just turn into a pile of liquid in our culture dish, but we want to actually make this into some kind of three-dimensional construct that's going to have the right shape and size that we want as well. So this is the focus of my research. How do we make sure that our bioinks will ultimately be printable, and how can we screen these so we're not having to make hundreds of different configurations with different concentrations of polymer solutions, but instead be able to just tailor the exact properties that we need so we can print our inks for our bioprinting. So let's talk about a couple of the different kind of conditions that could affect the printability of our inks. And so the first is regarding its composition. So the type of polymer we used, I mentioned synthetic or natural-based polymers, and also their concentration are absolutely key. And these can have massive impacts on the biocompatibility as well. But in terms of the printability of it, we want to make sure that these are all absolutely successful so that we're getting a nice, beautiful fiber that you can print into any shape and size, as you see here. And then cross-linking additives. So this is applying UV light or those different chemicals that you add that makes that bioink mechanically robust after the process. The second one is the printing operating parameters. These have a massive influence. What capacity does your printer have to be able to use different printing pressures so it can print very firm materials as well as very liquid materials? And so some of the factors include the printing pressure, but also the collector plate velocity. So how fast does the plate that's moving in a three-dimensional space need to move to be able to collect that material to make these beautiful shapes. And then also the needle size, which controls then how thick those fibers are. And the temperature control as well is really, really important. So these are all of the factors that we want to, that we have the ability to change and tailor for all of our different hydrogels, but how do we select the exact, the exact parameters of all of these so we've got something that is actually printable. So this is what I was looking at. Here is how we can assess bioink printability. How can we tell if a bioink that we're trying to develop is actually going to be printable? So the first thing, we want a fiber to come out of the bottom of our needle here. We don't want a droplet. And so this is what we look for first. And then our second key thing is we want to make sure that we can stack these layers. So this is a side view of our scaffold, for example. Make sure they all stack on top of each other nicely as opposed to all kind of settling down into a shape that's not desirable. And so we can screen for these particular properties really, really easily. It's putting your hydrogel into a syringe with a needle on the bottom and depressing it. And by hand, you're able to tell these two properties very, very quickly and easily. And so you can screen a range of concentrations or different formulations very quickly. But there are two slightly more complex parameters that we want to look at. And the first is cell suspension. So making sure that the hydrogel has the right consistency and viscosity so the cells are suspended in it and so they don't settle down to the bottom, and then the first bit that you print would contain all of the cells and then the rest don't have any more in them anymore. Or that it's not so firm that the cells can't then be extruded. And so a property of hydrogels is that they're shear thinning, and so this means that they get more liquid, so they get more viscous as you apply force to them. And so this is what happens through the needle. They start getting more viscous, and this allows the cells to move through the needle without being damaged, but then also be extruded into a fiber at the other end as well. And so the aim of my study was to characterize the yield point and the shear thinning behavior. So these are rheological properties of these materials, looking at their viscosity and their elastic behavior. Characterize these with some very simple, easy characterization techniques and be able to tell straight away whether our material will be able to be printable based on satisfying these conditions. And so this is just summarizing these, our yield point for cell suspension. And so we use our rheology measurements, which I'll show you in a minute, and then also our shear viscosity. So looking at how the material changes as you apply new forces to them to make sure that overall our material will be printable. I used a lot of different ink formulations, and so these ones are all very popular in the literature and are used by a lot of different groups for different applications. And they range in shape fidelity, so this is how well we're able to make some really beautiful constructs out of them, and also biocompatibility. So firstly, I used Pluronic, which is a synthetic polymer. It was originally designed for cell culture in very small concentrations, but in the concentrations that we need to be able to print it, it happens to be quite toxic, so that's why I've put it at this end of the biocompatibility spectrum. I used a pre-cross-linked sodium alginate, so now moving on to a natural polymer that we can cross-link with a particular chemical, and this forms reasonably biocompatible, but not overall very pretty hydrogel for our 3D printing. And then turns out Nivea cream, so your standard hand cream, is actually really, really great for bioprinting. So a photo I showed earlier was bioprinting with Nivea cream. It's got the exact right rheological properties that we want for bioprinting, and so we use this as our control material. We compare all of our other bioinks to Nivea cream to make sure that that's what we're going to be able to print with. So very high shape fidelity, low biocompatibility. Putting cells into Nivea cream is not such a good idea. And then I also used a blend of alginate and gelatin, so both natural polymers, and this gave me a hydrogel that gave me very good shape fidelity, but was also really biocompatible. And so this is being used all around the world for a lot of different applications because it's ticking all our boxes by the looks of it. So this was my first initial screening. I said we want to look for fiber formation and not droplets, and we want to make sure that we can stack it nice and, in nice pretty shapes for making our ear constructs or whatever we want. So here's our Nivea cream, and look, it gives us an absolutely beautiful fiber, and you stack it on top of each other in a cross and it doesn't start blending together. It's absolutely perfect. Here's some different Pluronic concentrations, and so you can see at very low concentrations these are droplets, And as you up them, once we get to 25%, here's our characteristic fibre formation. And also you cross them over and they don't blend together, whereas this one's starting to blend in the corners. And these are basically just liquid on a plate, so that's completely useless to us as well. So very quickly, we can see just by hand, by dispensing these, that we're getting our fibre formation and our layer stacking, which is absolutely critical. And then I also am just showing you results for different concentrations of alginate and then different cross-linking solutions as well. And so we found out that using 8% alginate with 1% of our calcium chloride solution gave us a really nice hydrogel that was able to satisfy those two conditions. So here's my beautiful bioprinter. This is my baby, and I was really fortunate to be able to use this over in Germany. It's a very expensive piece of equipment and really great to get some experience using it, which was awesome. So I could print with up to 5 bars of pressure. So this means I could move it around between, needing a lot of pressure to print some very, very firm materials, but also some very small pressures for very liquid-like materials, and a range of different needle sizes as well, so I could get fatter and thinner fibers. So here are my optimized structures that I was able to make with those different concentrations. So I decided that 25% weight pluronic gave me these beautiful structures. Here's Nivea cream, just looking stunning as always. My 8% alginate with the 1% cross-linking solution gave me a reasonably nice looking lattice, and then our alginate gelatin was our really biocompatible one, which was great. So these are all directly bioprinted. So let's get into the rheology. So this is looking at the fundamental material properties of how liquid-like or how firm these gels are. And we can do this by applying different shear rates or shear stresses. So this is applying forces to the materials and then measuring their viscosity with those different forces applied. So we wanted to look at the yield point. And so this is the material when you start to apply increasing forces to it, that it then starts to flow as a liquid. And this is really, really key for us to see if we want the yield point as this first stable data point in this particular flow diagram, and this tells us if the material will be able to suspend those cells or not. And then the other one was looking at the shear thinning. So as we ramp up that force that we apply to it, looking at how the viscosity decreases, so it turns more and more like into water as we apply more and more force. And so this slope here, this linear region, we can characterize with an equation, and we can start to be able to compare these with numbers as opposed to just looking at the graphs, which as someone with a physics background is very, very handy. So here's some data looking at the yield point limit. So remember, we're looking for these first stable data points when the material starts to flow. And you can see for that low percentage of pluronic here, the material's already flowing, it's basically water. As we upped that percentage, these yield points started increasing. So we can make a limit. So this is looking at the limit of the materials that could suspend cells, and then looking mathematically, what did that look like as a shear stress? And so we had 2 different formulations. One could be pipetted or suspend the cells, and one couldn't. So our limit has to be somewhere between 6 and 24 pascals. But then we can further define this because our alginate couldn't be pipetted, so it couldn't suspend the cells, but it had the limit of 9. So this tells us that inks that have this yield point, this particular point here, that's above about a 6 to 9 pascal limit, is unable to be pipetted and therefore unable to suspend cells. So that means you can take any hydrogel that's being developed, run this very simple measurement on it that takes about 10 minutes, and you can tell automatically without even having to put cells anywhere near it whether those cells will be able to suspend or not. And so if they can't suspend, maybe you need to do something chemically to that particular material to make sure that you are putting it within this limit. Then I had to do a little bit of maths, which made me really happy because I love doing some maths. And I promise these are the only equations I'm showing you, and I'm about to color code them, so it gets really, really good. This is where I'm looking at applying pressure and extruding our material through a needle. So this is a blown up version of what our bioprinters are basically doing. And I want to look at the average extrusion velocity. So look at how fast the material is coming out, which means how fast we have to collect it to get some nice fibers. Now I told you in the shear thinning diagram we could use an equation to model that. We get 2 nice little numbers from that, K and N, and so they pop up in these equations here all the time. But they're just fundamental numbers that we get from those graphs from the measurements, and so that's a really easy procedure for us. We also need the pressure. So this is looking at what is the pressure that we're applying on the bioprinter. Well, we've got a little nozzle and we kind of up and down that as we want. So that's our delta P, our pressure that we apply. And then we've also got the length of the needle and its radius. And so these are just fundamental to whatever needle that we've purchased for our bioprinter. And so what this tells us is that there's a very specific range in which the materials can be printed. So for the Nivea cream here, looking at some different needle sizes, and then different printing pressures up here. With very low printing pressures, this is unable to be printed. There is a window here where we're able to successfully extrude that material into the shapes that we want. And then there's a window up here where the material's just flowing out far too fast and we can't catch it in time to make some fibers. And so I applied this to all of the different materials that I screened to check if it actually works. And sure enough, let's look at the Pluronic, for example. In all these cases, this isn't enough printing pressure to get it. There's a nice window here where it works. And then here, the printing pressure would be far too high and we can't print structures anymore. And so I showed you those nice pictures of the results that I got of those structures. And it turns out that every time they fell within the bioprinting window. So we can now use this mathematical model, which is very simple. It's actually just an Excel spreadsheet, to be able to tell us automatically for any material whether there is a window within what our printers are capable of with different needle radiuses and also printing pressures to be able to tell if that material is printable. And so these tests take half an hour with the rheometer, and this mathematical model will tell us whether we can suspend the cells during printing, but also tell us about the shear forces within the needle and whether the materials fit within this bioprinting window. There are a couple of other very useful kind of mathematical results for this, and I'll just touch on them really, really briefly. The high shear rate, so this is the force that we're applying to the needles, and this also, to the materials, and this occurs in the needle. High shear rates at the needle walls. This is a really interesting observation. So this is looking at the center of the needle here, so in the middle, and then looking across the radius. And so the wall of it is out here at 140 microns. And so at the needle wall, we see this blue line of the shear rate increases towards the edge, which means there are very high forces at the edge of the needle and low forces in the middle. And this is what potentially could be killing our cells as we're trying to print them. So we need to mitigate this. So for materials that shear thin very highly, so they turn very liquid, we see these very, very high shear rates. But this is quite a different trend for materials that don't shear thin quite as highly. The other one we can look at is the residence time. So let's say I'm a cell and I'm passing through that needle as I'm being extruded. How long do I have to pass through those very high forces that could be damaging the cell and ultimately killing it? And so we can look at the difference for the materials that shear thin. These residence times are about 1.8 seconds. So that's all that's experiencing these high shear forces. But for other kinds of materials, these are up at around 18 seconds. So it means a cell is being exposed to these shear forces for a whole lot longer and ultimately could be reducing the viability of our constructs because we're killing the cells just as we're dispensing them through a needle. So we can see the difference here. More cells are being protected by these rheological properties as opposed to these very small ones. So only a small percentage cells are being protected, whereas all of these ones are being exposed to these very high shear rates that could be affecting the biocompatibility as well. So we always have to get to the end of our study and see, what have we done something that's reasonable, especially in applying a mathematical model to a physical situation. And of course, I only looked at shear thinning inks, and so these are inks with the very specific properties that get more liquid as you apply force to them. I've used a very simple pressure-driven extrusion model for 3D bioprinting. So there are, of course, far more complex bioprinters that apply cross-linking as they're being extruded. There are ones that have multiple axes of needles, so you're printing materials inside of materials that could protect them. I was just looking at simple pressure-driven extrusion of biomaterials, which is a very, very popular technique because of its ease. But we need to know that that could be a limitation of our techniques as well. There are also a couple of workarounds for this. There are other ways of cross-linking our materials to partially cross-link it, print it, and then fully cross-link it, or apply some different cross-linking methods as well. And so these all need to be taken into account when applying this mathematical model to new bioinks. But ultimately, we hope that with these two bioink printability assessment methods, we can characterize the yield point to tell if it can suspend cells or not, and we can check if it shear thin extrudes, which means that we're protecting our cells and able to make stable constructs that look exactly the way that we want to and have the right mechanical properties as well. So thank you. I hope I didn't completely confuse you all with all of that science. I'd just like to say none of this is possible without my research team, so I'm very grateful to our partners at the University of Würzburg, my supervisor, Dr. Jürgen Groll, as well as Thomas Jungst, and then also the Biofabrication and Tissue Morphology group here at QUT. And thank you to everyone who came to support me today, which is great. The Biofabrication Master's program was how I was able to do all of this amazing research, so I'm really grateful to that. and I'm now working as part of the ARC Training Centre in Additive Biomanufacturing during my PhD. So I continue looking into some of this stuff. So thank you.
You explained that so clearly though, like I actually understood maybe 85%. I don't know if I'm on myself by saying that.
85's good, we like
85%.
that.
So does anyone have one or two questions? Yep.
Quick question.
Yep.
Which type of cells you use? We have like induced pluripotent stem cells and different kinds of stem cells. So which stem cells you use to print the?
You can use a lot of different cells, and all over the world they use every type of cell you've ever thought of for this particular stuff. For my application, we were using mesenchymal stem cells, and so these are the ones that would ultimately turn into bone or cartilage. And so that was our goal, is to use these stem cells that would ultimately turn into cartilage for our cartilage applications. But you know, this is done for a whole wide range of applications, and so we hope that any group who's using any type of cells would be able to take this model and still be able to use it. Yeah.
Is it more cost effective to have a machine, maybe in the future, that cross-links as you print instead of having to do that whole process? Do you know what I mean?
It really depends on the application. So there are a lot of materials that it's a lot better to extrude them while they are thin and uncross-linked.
A purpose behind looking at them in different. Okay, that's
Yeah, absolutely.
Okay, good. Oh, Matt, do you have a question? Do you want to? No, Matt, no. You can't ask a question. That's the power I have as the chair. Okay, so next up we have Felix, who's going to take us through a scaffolding technique. And it involves something like an electrical whip. And then there's scaffolding in that. So it should be really interesting.
You actually said it all.
I know, so basically we can all go home.
Thanks, Naomi, first of all. Really, really good talk, and bringing a really complex environment where only you are the champion in as a PhD student into 10 minutes, 20 minutes is a great achievement. I won't be as good as Naomi as a starter. I presented this as my PhD last week, so this is the major content I'm going to talk about. What we as a group have achieved within the last 3.5 years. I'm very lucky that I have a, how do you say in English, a successor, like somebody who actually starts here. We are working together around here, but Joe starts here and keeps on going. This all goes into towards the bigger environments, the cell manufacturing industries. So I had fun. He will be working in very severe fields. Yeah, good luck. But yeah, again, thanks Naomi for actually presenting such a big content in such a small time. And also interesting, I think like this is one of the most important things we all have to do, because when I think back 2 years ago, Svenja and me, or I was actually invited by Svenja to join the BioArt projects, and I think if both of us hadn't been able to communicate what we do within 5 minutes, these conversations had taken ages. So we were really like bringing things on spot from both sides. And yeah, again, thanks for this opportunity, and also thanks to the panel and the invite here. What do you say, electro-whipping fibers?
Electrical whipping of scaffold.
Yes, perfect.
I am a doctor, so.
Hang on, I start this here. No, there we go. Yeah, cool. Design and development of an additive manufacturing technology platform for melt electrospinning writing. The electrical whipping is called electrospinning. It's a very old technique. We all, or most of the tissue engineering people know about this. It's all about applying electrostatic forces to viscous polymers to actually generate really thin fibers up to the submicron and nanoscale. We'll talk or get more into detail in a second. So we all know about tissue engineering, but I don't have to go into the, there we go, cool, definition. But for us, and especially in Dietmar Hubmacher's lab, most interesting is the area of 3D printing. Also what Naomi very well introduced with hydrogels. I guess everybody knows about 3D printing today in our society. It's all over the place. Two things, reproducibility and high throughput. That's the challenges we look at if we want to bring this from bench to bedside. Melt electrospinning writing, as I said, is one of these technologies when it comes to additive manufacturing. And with melt electrospinning writing, we combine, this might be the laser pointer, yeah, we combine traditional fiber or fused deposition modeling techniques. Also Naomi's project of the Bioink printing is part of this, where you extrude a viscous polymer, either heated or like sufficiently liquefied to a state, according to the movement of the collector or the relative movement to the needle, to create porous structures. On the other hand, we have fibers created with electrospinning. It's a very old technique where you apply electrical force to a viscous polymer, and then the polymer travels towards the collector but starts buckling really heavily due to the electrostatic force and ends up in this randomized meshes. So interestingly, if you compare this side and this side, is that we have ordered structures here, but a very large scale, and we have very randomized structures here. However, we have but very small scale, small dimensions. Melt electrospinning writing is a combination of both. Dietmar and his colleague Paul Dalton published a very important paper, which I don't show here, shame on me, in 2011, where we literally, or where they literally increased the collector to needle distance, decreased the collector to needle distance, and substituted the polymer dissolved in a solution with just heating the polymer. So it had a better viscosity, a better molecular stability to catch the fiber in a direct writing way. So literally, or at the end of the day, they started to be able to create 3D printed structures with morphologies in the lower micron scale. Again, the key features, what you can see here, we can now really deposit really, really small fibers very accurately, and can create large porous or highly porous constructs, which are really important for the tissue engineering community. We published this a while ago, beginning of the year. This is a graph actually showing a solid body up here, which is a centimeter to a millimeter, providing 240 square millimeters in surface area. Whereas when we reprint the same volume with a fiber diameter of 5 micron and a pore or a distance of fiber distance of, what is it? I think 100 here, we can reach up surface areas to up to 6,000 square millimeters, which is a, I think, 26-fold increase of what we have as if we would do a solid body. That is very important. Naomi introduced this. We want to get cells into the construct. So, and when there's a lot of surface areas, the cells can go in and attach and proliferate. Biocompatibility, yes, the material does not bear any solutions anymore. We only print with molten polymer, so there's no residual toxicity for the cells. And as you can see here, some examples how beautifully the cells actually align around these squared structures and eventually close the gaps. Exponential rise in publications as well as commercial interest, expressed by a decent increase of patent applications, and it's still going. As we see here, a lot of publications, and already we have initial interest from industry. So we have 3 medium to bigger companies worldwide who actually implement the technology in their existing technology platforms. Naomi, you can remember this one? This was the printer you used in Würzburg. And they have attached actually a melt electrospinning head recently. Really interesting. So yeah, if you look back to what when I introduced the fused deposition modeling or the solution electrospinning, if you compare where the technology from their side is now to where we are with electrospinning, we have a big gap, and that's where actually my PhD tackled. We don't really have a technological basis. I just realized, forgot to introduce myself. I studied mechanical engineering in Munich, so that was my topic where I started to think about how can we literally physically not get to these points of a really beautifully designed machine, but just finding a technology platform where you can approach something and just type in what you want to have in terms of yes, I want to print this object. You get the SD card in rather than just tweaking around with the parameters to get a perfectly aligned scaffold. 3 problems. We don't really have a technology basis. There's a lack of a lack of machine design. We have a really highly parametric process, again, as with the Bioinks. Here we have pressure, temperature, heat, collector speed, voltage, creating these, sorry, it's a bit small now, creating these fibers. And thirdly, what I also introduced in the beginning, the upscaling is one of the issues. So this is very interesting for medical applications. This is really efficient for tissue engineering. However, if it cannot get or cannot reach like a higher throughput, industry will never ever be interested in that. So I was lucky to be part of a CSC within the last years, where we first of all actually evidenced that it is possible. Joe will talk about this in more detail. Evidence that it is possible to use these lattices, surface treat them and grow cells very effectively. The project is about T cells. We published this and filed a patent to secure the environment. And yeah, so this, as I said, Joe is actually taking over, and he will bring more industrialization in this technology. So this was proven. There was the 3 gaps. Me, as I said, a background is engineering. We apply a lot of systems engineering and design approaches, systematic approaches to designing machines. And I hypothesized that when we more organizedly design a machine, we can gain more control, process control, and can actually can conduct more reproducible and precise research. I quickly just go through what we have achieved within the last years. So first of all, we did not just design a machine. We really thought of, or in a systematic or systems engineering design approach, we generated functionalities which have to be fulfilled. We conducted studies in terms of what is the perfect hardware. So we related, for example, the viscosity to the eventual system. And this is a very complex engineering approach to design perfect systems, which never are perfect, but it's more or less a good example for how effective interdisciplinary work is. Because when we designed this, we talked to the biologists, we talked to the electrician, we talked to the electrical engineers, we talked to all these people. What do you actually want? What do you expect of the system? And we brought it all together in requirements lists. This is systems engineering protocols where we just summarized everything and could find good connections between the individual parts. And this is how we started, and this is where we are now. We conducted and published experiments with these machines and can provide very competitive results. So we didn't build a machine which is completely crazy. We just build machines now which work all the same, all the way, all the time, and are reproducible. So that was very positive. Next topic then was how do we even more automate the process? And the process automation was not about using it, but rather about collecting data. That was exactly as in your probably your project, Naomi, when you have 5 parameters and you have if you just have 3 variations or levels of each parameter, you will end up in 243 different combinations. Manually, you cannot really do this. And in our case, it's pressure, temperature, speed, the voltage you apply, and so on and so forth to create this little fiber here. So we thought we cannot use an experience-based approach. We need to automate the whole process to collect a lot of data. Again, systems engineering very important to maintain or keep the overview. And then we built this conductive conveyor belt which was constantly running. One camera was watching the fiber here, the other camera was monitoring the diameter. Here we see it in real time. And we came to a data set which comprised 15,000 entries, which was too much even for our statistician team. They said, oh, we have to find a way to analyze this. But a good example, I think, for what we will be looking at in the future, especially in the medical fields, it's definitely big data and how we deal with big data. So the results were interesting and very positive. We now have evidence that when the fiber is stable, when the flight path is stable and doesn't fluctuate, we also have a very stable diameter. So this is very interesting for process control. We have made interesting studies which can now analyze 4 parameters at the same time, how they influence the process. We verified the results, so we could transfer the conveyor belt machine back to the machines we built initially. and transferred the whole thing to the first ever upscaled device for melt electrospinning writing. As I said, pretty much needed by the industry. We have a device here. It's actually in our lab, so I welcome everybody to have a look after this afternoon, where we have a machine. It can't be really seen here, but we have 8 printheads at the same time, printing on a larger collector blade, ending up in really precise scaffolds. We can repeat scaffold production as well, so we can print over 1,000 scaffolds in 3 days. But we also can print scaffolds which are almost a square meter now in size, and that is really interesting, especially for the cell manufacturing industry where we come to now. So that's how we envision the future. We got a lot of these machines stuck to each other. I want to thank again you guys for the invite and this nice panel. Thanks to Dietmar, the CSC, QET, and my home Munich. And that's where Joe starts now.
Unless anyone has a specific burning question, I think we might wait until after Joe's talk because he's going to discuss more about scale. And so since Felix talked a lot about scale and in terms of the machines, I think that we might be able to save our questions till the end. Okay, great. We'll just wait till the end of Joe's talk and then save up those questions.
Thanks for that introduction, Felix. Certainly done some great work as a preamble to what I need to do going forward. So my background is also in engineering, electronic engineer. That if you asked me about just over 12 months ago, you'd be at the university doing this sort of thing, doing a PhD research. I certainly wouldn't have believed you. But it's been fascinating because it's a passion of mine to be involved where technology and biology intersect. And this has been a project that certainly gives me the opportunity to do just that. So talk to you this afternoon about the challenges and opportunities in the manufacturing and expansion of cells for therapy. And I'd like to introduce that to you through this roadmap, where I'll be talking about the cell therapy landscape firstly, give you an idea of what's happening in the space. I'll move on to tying in with what Felix was talking about, the melt electrospinning writing, and we like our acronym. I'll ask the question as to whether we'll be able to draw some inspiration from other industries and whether this will inform the way we look with enthusiasm towards cell therapy and making it affordable. I'll give you a different set of views of the translation landscape, and this is the traditional, I guess, wording of from bench to bedside that often is used these days. And I'll give you a bit of a perspective in terms of looking forward in terms of my research. What are my next aims that I'll be going towards? And so as in Felix's case, I'm also being partly sponsored by the CRC for cell therapy manufacturing. And their remit is to bring cell biology and advanced materials together to address industry issues in a way that is collaborative. And so I'll start off by looking at what the World Health Organization did earlier in this decade, where they brought out this report looking at these non-communicable diseases. And their prediction is that in the current decade, this prevalence of these non-communicable diseases will in fact increase by about 15%. And this is evident by, if you look at the clinical trials, what did I do? When you look at the clinical trials and how they're focusing on these non-communicable diseases, a lot of them actually, you look at the bottom right-hand side of the graph, you can see there's a lot of effort being put into oncology, into genetic disorders, infectious diseases, cardiovascular, and so on and so forth. And the counter to that is, though, is when you look at the number of clinical trials that actually advance through the various stages. So you've got phase 1, 2, 3, and you actually get then to an advanced or an approved therapy. And that number comes down. And what's the reason for that? Is it because There could be an obvious reason that certain trials just take longer, and therefore, over time you'd get a reduction in them. Or is there another aspect about the actual approval, the support, the regulation that's around that may influence that? And so I'd like to just take you through a video with looking forward in terms of cell therapy.
Antigen receptor, or CAR-engineered T cells, can be designed to enhance a patient's immune system to target and kill cancer cells, independent of MHC on the cell surface. CARs consist of an antibody fragment that binds to a cancer antigen and signaling domains capable of activating the T cell. As part of this process, a patient's own T cells are collected and engineered to express a CAR gene specific to a cancer antigen. The CAR T cells are then expanded and infused back into the patient, resulting in an individual approach to treat cancer. After binding to the cancer cell, the CD3 domain of the CAR delivers the essential primary signal for T cell activation, termed signal 1. The co-stimulatory domain provides a critical signal, termed signal 2, to enhance the expansion, persistence, and potency of the CAR. The synergistic effect of signal 1 and signal 2 leads to an amplified effect. When the CAR T cell binds to the target cancer cell, it releases molecules which then induce the cancer cell to undergo apoptosis, or programmed cell death. CAR T cells proliferate, which increases the number of T cells that can recognize and kill cancer cells.
That's just an amazing technology if you think about what's being done at cellular level. So as recently as about 3 months ago, the FDA approved the first gene therapy in the United States. And this was a therapy that really was a last-ditch effort for a lot of these patients that nothing else was working for them. There's about 3,100 new patients every year that suffer from what's called acute lymphoblastic leukemia. This is just a disease that gives these young adults just no hope for life. But what's fascinating about this treatment is that 83% of patients are actually in full remission. So there's no sign of the cancer after being treated in this way. From my point of view, the interest is on the cell quantity, how many cells they need per kilogram of patient's weight. So for an average person of, say, 70 or 80 to 100 kilograms, you're going to need almost 20 billion cells. So where do you get these cells from? And this is where the technology that Felix was talking about earlier is instrumental as a vehicle to get us there. Now, interestingly, from an FDA point of view, whilst there's a lot of criticality in terms of how they approach things, they've actually certainly woken up to the fact that certainly something needs to be done to hasten the process, to make it more streamlined, to make it easier for companies to actually get these therapies through from preclinical phase to actual clinical phase and to approved therapy. And so they've had these 3 designations: orphan drug, breakthrough therapy, and priority review status. All 3 of these are designed to shorten that time frame for approval of the therapy. Aligned to the Novartis was the company that's actually driven the approval of this, or developed this therapy, is a company called Thermo Fisher. Now they are the guys that produce these microbeads or microcarriers. And again, the microcarriers are the alternative. They're actually the gold standard for 3D cell expansion development. And so there's a couple of issues in terms of their application that's not so good. And that's why we believe that the lattice structures or the scaffolds that we can produce through the melt electrospinning writing is certainly a competitive advantage in this space. So you can see from the graphs there that the number of cells produced over a 7-day incubation period is very similar. And so there's certainly a lot to be said for using the scaffold process and using a biocompatible and biodegradable polymer in the process. You're just ticking all the right boxes. And so just briefly, you've seen in Felix's presentation the slides, but you can see the absolutely almost invisible fiber that's running pretty much around there. The fiber size is in the submicron sphere, and this is really where we get the advantages of being able to depose or deposit a multiple fibers at a spacing of say 100 microns over that scaffold area, and thereby create an enormous, almost as you said, is it 60 times or 60 times an increase, or 26 times the increase in the actual surface area. And that's where, which the cells actually like. So you can actually functionalize the scaffold, and that means just we deposit some good food on the scaffolds, on the lattices, and the cells like to either be near them or to adhere to them. And so the question, as I posed earlier, is from bench to bedside. If we had to get an order tomorrow for 10,000 scaffolds of, say, a size not the small centimetre by centimetre, but say 15 centimetres by 15 centimetres by 10 layers, it'll take us a lot more than a month to produce those. In fact, we'll be in a lot of pain, not literally, but it certainly will take us a long time. And so looking at the landscape for challenges in cell therapy space, we do see a lot of papers in the scientific field saying, we've got heaps of challenges here, things are not as smooth as we'd like them to be. We've got a new paradigm saying we've got to start doing things differently. We've got large-scale biomanufacturing challenges and opportunities. And then there's some pointers in the article space that says, there's probably there is potentially a way we can attack this challenge and get some answers. And equally, the engineering bodies have been looking at with interest into this space of life sciences. And a few years ago, there was this Grand Challenge conference where they acknowledged that there's a lot of knowledge and ability that's been developed over time. There's the application through engineering methodologies that's certainly helped us to understand the intricacies of cellular mechanisms. But have we seen a reduction in biomedical and health problems? And I'm not sure that is in fact the case. So there's a belief that optimization surely is something that needs to be done. So that area we've highlighted there really needs to be a lot larger. So there's certainly room for improvement. And so they've identified these 5 grand challenges, and the 2 ones in red are really the ones that sit close to my particular project. And so it's certainly inspired me to look at then what in other industries is actually potentially could help us when we look at cell therapy going forward. And I've drawn the analogy between semiconductor industry and cells, and we all know, we've seen what phones have become more functional. They've become, and that's largely because of what we've seen happening on this curve here. Way back in 1970, we used a feature size of 10 microns. Right now, we're working at 10 nanometers, and it's certainly going to go through to, they're already working on 7 nanometers. What that all means is, in fact, that they're able to pack more and more functionality into smaller and smaller bits and pieces. And so when we're looking at cells and we want to proliferate cells, we want to have more and more cells in a particular space, it certainly, there's a parallel to me that I certainly like to draw on. And so the obvious question then is, okay, so what happened in the semiconductor space that we could potentially draw on in the cell therapy space or cell expansion space? And so there's two classes of drivers, you can say, technological and economic. So in the semiconductor space, we had, as I talked about, the chip area and the feature size, improved design. But if we look at what's happened in the cell therapy space, we've had the deciphering of the human genome earlier in the century. As a result of that, there's a newly identified receptors and enzymes and transporters and ion channels. The whole suite of omics has revolutionized how we look at these challenges. On the pool side, we've looked at the emerging therapeutic market. People have seen the benefits, they've seen, and they want more, they want less, reduced side effects, and of course the prevalence now of using biologics in living cells. So this approval of the Novartis therapy has really been revolutionary. And so just to summarize, so we've had the FDA approval of Kimberly and Yescarta cell therapies, very similar cell therapies. We've looked at the WHO report. We've seen Moore's law in the semiconductor industry is really almost just as applicable as drivers for cell therapies. We've seen what the engineering fraternity are looking at integrating and collaborating in terms of the life sciences. And we've looked at the cell therapy landscape, the challenges and opportunities. Now, what does that mean? So I've tried to draw this picture for myself. And, you know, when I saw this for the first time, I said, this just, you know, it sort of boggles the mind. There's just too much there to focus on. But then you need to distill, you need to take a step back and say, okay, so we're looking at technology and infrastructure, manufacturing, regulations and therapy, and it's sort of pointing to a way in which one can actually then start to focus. And so it's led to the paper that was published earlier this year, and the highlights from that basically the landscape of challenges and opportunities for cell therapy. Basically highlighted a few points. Firstly, that there was a laboratory-based cell culture methods that were expensive for a number of reasons. You're using highly skilled people to do sometimes menial jobs, but need to have that skill set there. There's not necessarily an optimization of media usage and growth factors, so that leads to more costs. There's opportunities there for automation. There's the drivers, as I said before. There's certainly a desire and a lot more talking about collaboration. Collaboration not only between different engineering disciplines, but across disciplines. So you're looking at people with I guess, more of an artistic background, people with more of a scientist and engineers, and everybody's talking to each other, because within all of that talking together and collaboration, sometimes you get really good answers. And so good laboratory practice and good manufacturing practice often seen as the underpinning of any desire to manufacture in a reproducible manner. And that sort of becomes the baseline for any work in that regard. And then the belief that the 3D lattice structures, which Felix has now given us a few examples of prototypes that work really well. And so we can predictably, with all the data sets that have been collected, we can now predictably produce a lattice with the right kind of porosity, with the right kind of fiber diameter, with the right kind of spacing of the fibers, which is really exciting, because we know through all of that we can take the next steps to look at how do we scale this technology. And so this is another view of the mind map that I showed you earlier. So you can divvy it up into regulatory requirements, and you can see I've indicated some gaps, and there's gaps in the way that the FDA and the EMA in Europe and the TGA in Australia are coordinating things. We're not talking the same language yet. So there's certainly a few areas where effort needs to be put in to get the different parties to talk to each other, to understand the same language and start working together towards the objective of having an affordable business model, and a business model that closes that affordability gap to patients getting the essential health care. And so when you look at getting from bench to bedside and trying to get across this valley of death, you talk to any startup company, there's a lot of stories that they can tell you about what happens here. And I believe we can cross that bridge if we give enough focus to the gaps that I've identified in these areas. For my own research, I need to look at the transition from or shall we say the focus certainly needs to move from 2D to 3D models. And whilst there's some advantages in the 2D space, it certainly points all towards 3D models. Thank you. And so just to give you just sort of a still of the video that we saw earlier, what we saw in the video was essentially what happened in step 7. There's all these other steps that happen around it. So you've got the harvesting of the cells, you've got transport to some place where they actually expand the cells, they reprogram them, and they quality check them. Some transport or logistics again. There's something preparation done to the patient before you get cell infusion. So just to get an idea. So my interest really is in what are the underpinning technologies in this space. So I'll finish off now. Again, just looking at the technologies and where we need to be. For my research, I'll be focusing on typically this arm or this leg of the technology that's affecting and trying to ascertain the I guess the platform structure. not only from a technological point of view, but also from a manufacturing point of view when you're looking at scaling up or scaling out, looking for going forward. And so for my own research going forward, it's firstly the hypothesis is that using quality by design and such systems engineering approaches will facilitate a pathway towards studying proof of principle cell therapy concepts. And so the aim one is pretty much now done. My next step is to focus on the framework for cell therapy manufacturing using the melt electrospinning writing as a platform, and then as a final step toward developing an economic model, economic optimization model for the same. So it's all about getting to a point where we can translate this exciting technology to a level where we can benefit more patients and industry. So again, thank you to my supervisors and colleagues and for the opportunity, and also for the opportunity today to talk to you. Thank you.
So it's 4:30. I nailed it, I know. But if there is a question before we finish today, that would be good. Does anyone have any specific questions they'd like to ask? Matt, I will let you ask a question.
Just to loop back to the beginning of the day, we kind of started off with Nicole and Nelson kind of talking about patent law and 3D printing and bioprinting and some of the opportunities and challenges. I wonder what the thoughts of the scientists are in terms of navigating this territory yourselves, both as inventors of new research, but also users of patent material and perhaps experimental users. I just wonder what your thoughts are in terms of using the patent system. In the last talk, there seemed to be some attraction to other models of innovation in terms of grand challenges and prizes and other models of encouraging innovation. So just to tie things up, I wonder whether you'd like to reflect back on what some of the lawyers have said.
And don't forget Naomi behind you. She's re-entered the crowd, but we need her voice in this.
But I think,
Shine.
like one of the key aspects we have to like mention in this regard is it's one of the main questions, essential questions. I think we should all as researchers address to ourselves. But interdisciplinarity and working in a surrounding where I don't understand what you do, but I have to incorporate what you do. I try to understand what you do, and we have to work together, and so on and so forth. Like what we do on a technological perspective is not just engineering. There's chemics, there's biology, there's electrical engineering, kinematics, blah blah. There's a lot of things. So I think that I might bounce the question back to the lawyers and say, hey, so we just don't patent or put intellectual property on this one screw as it was traditionally done for 200 years. This is a complex system. We need more protection in a way.
My thoughts are that we're sort of in a bit of a conundrum in the one way, because as academics we want to get the word out there. We want to actually collaborate with people. We want people to know this is what we found. But in the space where we've got potentially intellectual property sitting there, we actually have to look after ourselves in a way too. So there's a fine balance that needs to be struck there, where we at some stages we probably need to hold back on publishing some of those results so that we can get the benefit of getting our IP protected first before we then publish. And
Which would stifle, do you think, innovation in the area if you had to hold back because you're waiting for some kind of IP approval, or?
I don't know whether that would stifle it. I just think that if you think about the advantages of collaborating on a wider front with more people, probably it will slow things down a bit. So stifling maybe is one way of looking at it. I don't think it will. So it's just a different way we have to look at it. Whether there's a way around that, I'm not sure, but certainly, and that's why I think it's important to get the legal side involved in that discussion so we can work together. Is there other ways in which we can protect ourselves but still get enough collaboration around and going so that we can get the maximum benefit of this effort?
Naomi, do you have something to add?
Just echoing what Felix and Joe just said, I think, as in a biofabrication research group, we've got very good at being a multidisciplinary research team. We have chemists and physicists and mathematicians and engineers and biologists and everybody coming together to solve these challenges on a scientific level. But it's working, you know, not only just multidisciplinary now in our research group and coming up with the language to communicate there, but also, you know, it's platforms like this where we can then share, you know, our perspectives and hear the perspectives from, you know, all of the other fields that all need to come together to make all of this possible. That's absolutely crucial. So yeah, it's events like this, and it's starting those conversations that, you know, we're really able to make a difference.
Yeah, and you're really good at like accessible communication, so that's going to be a real benefit for you in making that transdisciplinary. All right, Matthew has charged me with closing the event today, and I don't really have anything except lots of different thoughts, lots of different things coming out about hype, about the role in getting the balance right in IP. So we'll carry these conversations forward. So thank you for joining us today, and thank you to Matt Rimmer in particular for planning this event, as well as Ilana and Sharon, which is our admin team that really helped us pull this together, but mainly it was Matthew forcing behind the team that got us all together today. So thank you.